is Lana Daoud, and I'm a PhD candidate at the UAEU. And here I will share some of our recent findings where we focus on the emerging resistance to cefiderocol. Cefiderocol is a cephalosporin that has a siderophore side chain, and that means that it can bind iron and hijack the iron receptors on the outer membrane. Cefiderocol has shown remarkable potency globally. However, we continually see increasing reports to its resistance. And the traditional mechanisms of resistance of beta-lactamases can only partially explain these trends. So we hypothesized that there are other pathways that can be involved in the mechanism of resistance to the drug. Here we hypothesized that mechanisms or pathways involved in oxidative stress response as well as iron regulation can play a significant role on the activity of the drug. So in this study, we collected 100 Klebsiella isolates from clinical samples, and we tested the minimum inhibitory concentration, or the MIC, to cefiderocol under iron-depleted conditions. These samples were also sent for whole genome sequencing, and the assembled genome was specifically screened for amino acid variations in genes that relate to oxidative stress, iron regulation, as well as other antimicrobial resistance genes. Now, these mutations were then statistically correlated to the susceptibility patterns of cefiderocol. In this slide, in these figures, we can see the results. So first of all, we see in the green part that actually cefiderocol maintains its very strong potency on Klebsiella pneumoniae that are multi-drug resistant and extremely drug resistant, with only 9% being non-susceptible. Now, although this is a very small number, but it's very important to explore the causes of resistance to prevent this number from increasing further. So when we look at the amino acid variations, we were able to identify over 300 amino acid substitutions in these genes across all of the tested isolates. And these genes belong to the OxyR regulon, the SOX-RS regulon, iron uptake genes, as well as a flex pump. Now, each of these amino acid substitutions were correlated with the susceptibility patterns of cefiderocol. And we found that mutations or variations in the genes related to OxyR regulon showed a negative correlation to the cefiderocol MIC. On the other hand, genes belonging to SOX-RS didn't really show any correlation with cefiderocol susceptibility. Now, the important thing is that the iron regulation genes, specifically iron uptake genes, showed strong, significant positive correlation with the MIC of cefiderocol. This means that as the frequency of these substitutions increase, so does the MIC of cefiderocol, lowering its efficacy or its activity on these isolates that harbor these mutations. As for the flex pump, it also showed it didn't correlate with the susceptibility to cefiderocol. Now, we were also interested to see whether these patterns correlated with the clonality of the isolates. So here in the core diagram, we see that among the amino acid substitutions, the iron-related genes, they strongly correlate with ST14. ST14 is classified as a high-risk clone, and it's actually very prevalent in the UAE as well as the region. We also see similar patterns with other high-risk clones. So altogether, this shows that there is a complex interplay between the genetic variations in oxidative stress, iron-regulating genes, and the susceptibility to cefiderocol. So in conclusion, of course, we maintain the efficacy of cefiderocol, but we propose that the mechanisms of resistance are related to, let's say, unconventional pathways that are related to oxidative stress as well as iron-regulating genes. And of course, functional assays must be performed to validate the mechanistic activity of these mutations on cefiderocol. So that's all, and I thank IMME for inviting me to speak here today, and thank you very much.